Welcome viewers. Today the topic of discussion is transposons and transposition in prokaryotes. Now the outline of my lecture is introduction, then I will talk about discovery of the transposable elements, then the discovery of the transposable elements with respect to prokaryotes, then types of the transposable elements which are found in prokaryotes, namely the IS elements, the transposons, the Bacteriophage Mu, then the mechanism of transposition, and lastly we will discuss about the effects of transposition in prokaryotes. Now coming to the transposons, this term transposon means an element or a portion of a DNA which can move around. Now, and transposition is a mechanism by means of which it can move around. And due to its moving around, it can cause disruption of genes, or it can even not cause disruption of it, just move around to a region where there are no active genes or no housekeeping genes present. So the effects of transposition may be visible or may not be visible. Now, and why it is so important, we see that in the whole world, in the biological world, there is huge quantities of DNA present in certain organism, but the complexity of organism is not there. This nowadays can perfectly be explained by the theory of transposition and the presence of transposons. However, the discovery of transposon dates back to around 1940s. This work mainly started, the background started in the lab of Rollins Emerson in Cornell, and at that time they were working with maize or the common corn. And the importance of maize is because they have a large packet in chromosome, they can do squash and see the genetic details. The first traces of such discovery dates back to 1938 when Marcus Rhodes, he was working on different genotypes of maize, found a variegated condition and what you are looking at. So this is stunning, and some of us have come across such mazes, and we always think about it, how does this happen? But what is the genetic basis? And at that time also they were perplexed by why such variegation happened. And there were initial studies and mostly genetic studies at that time to see the basis of this variegation. Now pioneering work came through works of Barbara McClintock. So they all emerged from Emerson lab, and she, while working with this maize, found that for certain regions of maize, and I should tell you that maize has 10 chromosomes, 1 to 10, of which the first is the biggest and the last, that is the 10th, is the smallest. And she found that certain regions of chromosome 9 were breaking. And so the experiment she did is as follows. So what she did was she was looking at various traits. The traits were the kernel, that is the seed, and how the seed looks like, the shape of the seeds, the color of the seed, the layering, that is, and whether the seeds were shiny or they were shrunken. So these were controlled by 3 different genes. So she did experiments where she crossed with the 2 strains. The first strain was colored and it was bulbous and waxy, and the recessive strain, which was not colored, that is colorless, shrunken, and not shiny or not waxy. Now because they were located on the chromosome 9 and a particular region of chromosome 9, so she expected that the progeny will still have the dominant phenotype or the wild type phenotype. However, quite surprisingly, what she found was the recessive phenotype was getting expressed in certain region of the seed. Now what she hypothesized at that time was this was due to chromosomal breakage, and she attributed 2 factors due to chromosomal breakage. One, she called the dissociation element, DS, and she thought that this dissociation element was present at one end where these genes were present, and because of the dissociation at the DS site, there was breaking of the chromosome, giving rise to the phenotype which you saw. And you can see that here you have the seed with the color, and this part is the colorless. So large patches of colorless, and this colorless was formed due to breaking of the chromosome.
So if DS elements are mobile elements, then what will happen if it actually move around?
that was the experiment she did. And so what she found was, if you look at this figure, if the DS element was present, let's say, at a region or in the gene which is responsible for the colored kernel, and there were derivatives which were found, so that kernel will be colorless. The other features, so there is no breakage of the chromosome. The chromosome is there, so you still get a waxy, you still get a plum seed, but it's a colorless seed. So that, and it was because the DS element was present there. Now, coming to your question, if it moves around, then it will be a reversion of the phenotype, and that's what she found, and that was the explanation for variegation. So when it moves out, let's say if it moves out from this side, then the colored gene again expresses itself, so the coloration comes. If it happens after fertilization in the mature tissues, then only the tissues or only the cells which are derived from this reversion will show the color, and that explains this patchy appearance or the variegated appearance on the background of the colorless seed. And she attributed that it is not only the DS element but the AC elements, which are the activator elements, which required to be there to activate this movement of the DS. And unfortunately, she could not specifically map the DS or AC to a particular locus, and these findings led her to believe that these are mobile genetic elements, though the world at that time did not believe her. So the discovery of the mobile genetic elements, which is very important, and Barbara McClintock got recognition of the discovery almost 2 decades later, when the presence of mobile elements were also detected in E. coli and later on in organisms like Drosophila and Snapdragon, and nowadays we know that it is present in various organisms, and she was awarded the Nobel Prize in 1983 for the discovery of jumping genes. Now coming to the second part, which is the discovery of mobile elements in prokaryotes. Now, first and foremost, people wanted to look at the galloperon, which controls the breaking down of the different galactose sugars, and they found that sometimes there were mutations which were happening in the galloperon, and they were also reverting back. Now, they at the time mass scale sequencing was not there, but molecular biology was coming at its very babe. What they found was there were certain segments of DNA present in the galloperon, and these were disrupting these galloperon. And these DNA had a very important thing, which are the inverted repeats. So they were, as you look at the figure, so there are inverted repeats present on 2 ends, and this stretch of DNA was first called the IS element or insertion sequence. And the IS elements in E. coli especially, there are many IS elements, at least 8 of them are IS1, then there are 5 IS2s, and there are less known IS elements, and nowadays we know there are lots of IS elements which are present. Now, how people know about that IS elements get inserted in a DNA? So the experiment they did, and they took advantage while doing of a particular fact while doing such experiment, was that the lambda fudge get incorporated near the galloperon, and they can disturb the galloperon. So they took such strains, which is known as the lambda D, that is deleted galloperon strains, and they took out the DNA, and they wanted to separate this DNA from mutants of GAL4 or the GAL locus. And what they found was that when they took out the DNA and separated DNA using cesium chloride density gradient centrifugation, a method commonly used to separate DNA, and then they compared the length of such DNA or the density of such DNA with that of the wild type where the galloperon was not disturbed, they found that there was an increase in the size in the mutant strain, suggesting the movement of the IS element into the galloperon actually adds subsequent amount, and this varies from 800 base pair to about 5 kb to the galloperon.
Sir, are the IS elements just random inverted DNA sequences, or do they have any specific sequences?
Very good question. And that was the thing they also wanted to find out. So what they did to get the answer to you, was they did hybridization experiments. They took they extracted the DNA from the lambda gal operon deleted phases, and they used the DNA to make RNA, but they incorporated radioactive probe for proper identification. Now, when they hybridized this RNA with DNA from different such strains which were mutant in the gal operon, they found that it was properly hybridizing to such DNA, suggesting that they are not just random fragment, but there is some commonality between these stretches of DNA. However, if they took the original gal operon, which were not mutant, this DNA was not getting hybridized to it, or the RNA, the radio-labeled RNA was not getting hybridized to it, and which also suggested that the IS element was specific and they get incorporated into the gal operon. Now, coming to the types of the prokaryotic transposons or mobile genetic element, they can broadly be categorized into first the IS elements, second the prokaryotic transposon, better known as the TNs, and the Bacteriophage Mu. First, I will try to come to the IS elements. Now, the IS elements, as I have told you, they have the inverted repeats at its 2n. And within these inverted repeats, there is a chunk of DNA which codes for the transposes. So, these itself form a self-sufficient unit. They code for its enzyme, which help recognize these sequences, cut them, and move to other sites. So, the transposes is an enzyme which helps in cutting and moving away to different sites, and this is present in the IS elements. There are various IS elements discovered today: IS1, IS2, IS10R, 10L. These are few examples. The second type of transposable elements found in prokaryotes are the transposons or the TNs. The TNs are broadly categorized into composite and simple transposon. First, I will explain you about the composite transposon, taking the TN10 as an example. So, the TN10, if you look at it, it has the IS elements, just like the IS. It has inverted repeats at its 2n, and then they have certain other genes as well as the transposes gene. The difference between the IS elements or IS-like modules, which we just studied, and this transposon is that the IS elements here does not have its own transposes, or they are mutated. So the transposes gene is present somewhere in the middle, and as well as there are some marker genes, for example the tetracycline resistant marker, as you can see. There are various such complex or composite transposons which are present, which have different antibiotic resistant genes. The second type are the simple transposons. Now, the simple transposons, as you look at it, the best example is the TN3. They also has the transposes gene at the central region, as well as there are genes which help in the transposition mechanism. Now, if you ask me how these simple transposons are different from the composite transposons, so unlike the composite transposons, they don't have so many marker genes present. Now, the advantage of the composite or the simple transposon is that, especially the composite, is because of the presence of composite transposon and because of the shuttling around between the bacterial chromosome and the plasmid DNA, there are plasmids like the R plasmids which have acquired different antibiotic resistant genes. Now, I should tell you that both the composite and the simple transposon lead to target site duplication.
Sir, what exactly is target site duplication?
Yes, very good question. If you look at this picture, so let's say this small part is the stretch of DNA which is present in the target DNA. That is where the transposon will come and sit. Because of which, as you see, there is duplication. So what you are seeing in green, the duplication of this target site where the transposon has come and sit has happened. So this is called target site duplication, and in most cases it happens when transposon come and sit at a particular DNA locus. Now, coming to the third category of prokaryotic transposon is we should mention about the Bacteriophage Mu. Now, these are phages which can infect both in the lytic or the lysogenic stages, and they can incorporate their DNA into the bacteria, and this incorporation of DNA happens because there is some particular gene called mu-A present. Now, we will look at this portion of the DNA. So, this is the head, there's a tail, and this is the total mu genome. Now, what happens is there are particular stretches of the mu genome which cuts from the phage and get incorporated into the bacterial chromosomes. And again, it can shuttle around, and people can use it as a vector for moving around different bacterial genes or different large chunks of DNA using the Bacteriophage Mu. Now, coming to the mechanism of transposition, so we have studied too much about the types, but how is it actually moving around? I should tell you there are two broad mechanisms. One is the conservative and second is replicative. So coming to conservative, as the name tells you, what it means is that the amount of DNA is not duplicated. So what happens in this is the DNA cuts from one region, it moves to a new region, and that old region is empty. First, a double-stranded break happens, and this break is created by the transposase, which is the enzyme. And this double-stranded break is not a blunt cut but a staggered cut. So 3' OH groups are still present, overhangs are present. A similar break also happens at the target site. Now these transposon, or the genetic element, moves from this region, let's say locus 1 to locus 2, get inserted, and then there is transesterification reactions and sealing of the gaps, which can be done by ligases, due to which it sits into its new locus. And these types of transposition mechanism is exhibited by members like TN10, TN5, etc. The second, which is widely acknowledged method, is the replicative transposition. Now the name tells you that the amount of DNA is duplicated, or what it leads to is the increase in copy number of the transposons present. Now what happens in this mechanism is first the donor and the target plasmids or the DNA come near each other. One good example is we can take the TN3 as a very good example. Once it comes near each other, first there's a single-stranded cut in the donor side and a double-stranded cut at the acceptor side or the target side. Then there is a 3' and the 5' linkage between the donor and the acceptors or target DNA. So, and still there are some overhangs lying. These are then taken care by the replicative machinery, and it replicates it. From cutting till the joining, these are done by the transposase. So it forms an 8-like structure which is known as the cointegrate. The cointegrate is one of the most important intermediate in the replicative transposition mechanism. Now, is it stalls at the cointegrate? The answer is no. What happens is, then there are enzymes like resolvase opens up, and then there is again cut to open this up, joining of the DNA, to you now have two independent circles, and in both the circles you have portions of the transposons which is there. So that means we now have two copy from one left at the donor side, one still there in the target side. And these types of replicative transposition is present in most of the prokaryotes, even in the mu bacteriophage, in TN3 and etc.
So, how do you know about the genes which helps in replicative transposition?
Yes, very good question. So the way to detect, or the experiment to detect the genes which are involved in replicative transposition was done by mutagenesis studies. So what people do, they took out this DNA and they utilize DNAs to chop off small fragments and then incorporate it again in corresponding vehicles or plasmid, and then put them in the cells, and where some regions of this transposition were still kept intact. If you see the figure, and I can take a very good example of the TN3. So you see that genes are like TNPA, TNPB and BLA are present in the TN3, which are transposase, resolvase and lactamase. But how we know that these are involved? So let's make a mutation at TNPA. Now what it does is, if you make a mutation at TNPA, it never forms the co-integrate. They recognize the target site, but the cutting of the donor site and joining with the target site does not happen. It stalls and it can never be complemented. And because the transposer helps in cutting and stuff, so we now know that this TNPA codes for the transposase for the transposon TN3. The second is, let's say, the importance of resolvase. Now people again made mutations of the TNPB gene, and they found that the co-integrate was forming and there were lots of co-integrate accumulating, but then it did not resolve into two separate structure. And it can be complemented, it cannot be complemented depending on whether we put back the same gene in the same place or not. Similarly, the activity of beta-lactamase or various markers could be tested out. So at least the importance of the transposase or the importance of resolvase in the replicative mechanism could be understood by such studies. Now coming to the effects of the presence of these transposition mechanism in prokaryote, it can lead to DNA rearrangement, and we know that it can do depending on recombination, because the sites which are the IS elements, they can recombine between themselves. They have homologous stretches, though, and certain cases inverted repeats. So they can lead to rearrangement of DNA. They can lead to movement of DNA from one locus to another. Then it can lead to deletion. Let's say genes which are present in between two transposons or in between two IS elements, because of the recombination of the IS elements, can get lost. They can also lead to excision of genes. Let's say the transposon was sitting just behind or before the gene. Now as the transposon moved, it took a portion of that functional gene also away with it. In most cases, precise excision, that is the total gene moving due to the movement of transposition, is rare, but imprecise excision happens. Then it can lead to fusions of episomes. The best example we studied is due to the F factor, which integrates into the bacterial chromosome or the bacterial DNA of E. coli. Then fusion of large chunks of DNA, and this is a very important application of Bacteriophage Mu, because in conventional molecular biology method, we cannot move around large chunks of DNA, but we can utilize the Bacteriophage Mu. Because of the presence of mu A, we can put the gene of interest or the chunk of DNA in interest within that transposon, and because of the movement of transposon, the whole chunk will move to a new locus. That there can be some polar effects. What I mean by polar effects is, because of the arrangement of the transposon and where is it sitting, either in front or in back of the gene, it can influence a function of the gene. Let's say if it sits in the middle of a coding sequence, then it will disrupt or it will inactivate the gene. But if I tell you that transposons only cause gene inactivation, it's totally wrong. In certain cases, people have found that transposons like IS2, if it sits in the promoter region of certain genes, or if it sits just at certain regions in front of the gene, it can lead to activation of the gene because of the read-on mechanism, though the exact mechanism of how this activation of certain genes happens is still debated. To summarize what we have discussed today, we have today discussed about the discovery of the mobile genetic elements or the jumping genes or the transposons, and we attribute it to Barbara McClintock, for which she got Nobel Prize. Then we have discussed about the discovery of the IS elements in prokaryotes, which are the first prokaryotic transposons reported. Then we have taught about different types of the prokaryotic transposons, namely the IS elements, the TN elements, there can be composite or simple, and the Bacteriophage Mu. We also discussed about the two major modes of transposition, the conservative mechanism and the replicative mechanism, and what are the effects of such transposition mechanisms in prokaryotes.